The process begins when acetylcholine from the axon terminal, represented by green beads, is released into the synapse. The acetylcholine travels across the synapse and binds to receptors on the sarcolemma. The binding of the acetylcholine creates a new action potential. The action potential spreads along the sarcolemma. The action potential travels down the T tubules, which are associated with the sarcoplasmic reticulum. The sarcoplasmic reticulum is the blue tube or branch-shaped structures, and the T tubule is in between. This action potential affects the terminal cisternae of the sarcoplasmic reticulum. It causes the terminal cisternae to release stored calcium ions, the pink beads, into the sarcoplasm, which happens within a millisecond. The calcium ions then look for troponin to bind to. While this is happening, the ATP, the blue button that is on the myosin head, splits into ADP, the red button, and P, the white button. The ADP and P remain on the myosin head. The split energizes the myosin head to prepare it for attachment to the actin. The calcium then binds to troponin, causing the troponin to change shape. The troponin pulls on the tropomyosin, exposing the previously blocked binding sites for the myosin heads to attach onto. Myosin then attaches to the actin, creating cross bridges and releasing the phosphate group. Finally, the binding of the myosin head to the actin triggers the power stroke. During it, the myosin head swivels and releases ADP. The myosin head pulls along actin and moves the thin filaments towards the center of the sarcomere, the basic unit of muscle fiber containing actin and myosin, causing muscle contraction.